Alright, y'all, we're gonna do this test prep. Austin is playing Animal Crossing and doesn't want to leave. Are you gonna leave for me, please? So I can do this, so that we can do something else? Can't squish you. Squish, squish, squish. That's what I was doing. Hey, Austin, can you leave though? Austin, you can catch a clownfish and stand up.
Let me catch clownfish first. Swimming toward me.
Today is sponsored by Dove. Life is hard. Okay. Today we're gonna do the nervous system portion of the test prep. I'm gonna post it, and then tonight at session time, I will be up for Q&A and everything. And then again on Thursday, same thing. I'll post the session that's recorded of the cumulative portion, post it, and then be up for Q&A. And then on Friday we're done with SI. I know y'all's final is soonish, and I want y'all to get the most out of everything. You have the website, you have YouTube, you have the playlist I have on YouTube, you have Lauren's stuff, you have Quizlet, you have literally, there are so many things. I believe in you. You're all going to do great. Finish the semester strong because we want to do something besides school, right? We want summer and stuff. But, so I'm going to go get my water and let's do this thing.
It's called equivalent exchange. Watch Full Metal Alchemist. Then at me. You had a jump.
Go away, please.
I'm leaving this room.
Correct. Okay. This is the nervous system portion, and we'll do the cumulative portion, right? Both of these reviews are on the website already, so if you wanted to have filled them out or do that beforehand and then check yourself, or vice versa, or do it afterwards again as like a review thing, whatever makes you happy. Okay. Zip pages. Let's do this because I have other things to do. Also, session 17 is up on the website. I put it up, or on the YouTube. I put it up like 6 or 7 hours ago. It's not that long ago. Sorry about that. It's just like I just didn't want to. I recorded it last night and whatever. Okay, so a lot of this is going to be literally review because we talked about it. So bear with me, but this is more of a we're enhancing your knowledge thing. This is what I want. Stop being angry. It's angry at me. Okay, so we have overlapping functions of the nervous system. Sensory input, right? We're sensing something, right? We're picking up the glass. We're sensing how this feels. We're sensing that it's cold. We're sensing that it's heavy, right? Integration, right here, right? Where we're actually understanding and interpreting what that information means. So this is just a stimulus without me thinking about, oh, maybe how does it feel? Is it cold? Is it warm? There's no integration. There's no thoughts to have me describe what's going on. And then we have motor output, right, to the effector organs or to the muscle, right? So we feel this, we feel it's really cold or really hot, and we want to put it down. Motor output is I wanted to put it down, okay? Organization of the nervous system is huge, 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 huge. Go over this. There's videos about it. You could even make this, or it's blank on my website. Do this so that you know where you are in the organization. If you don't know, then you're kind of in a world of hurt here. Okay. Major division: central nervous system, brain and spinal cord, right? The control center, right? And then we have peripheral nervous system. Okay. So these have the separations. This is nuclei and tracts, and ganglia and nerves. This has the terminology that you need to know on here. So for the central nervous system, we have nuclei, which are clusters of neuron cell bodies, right? However, in the peripheral nervous system, they're called ganglia. Same thing, different organization, right? And then we have tracts, which are the bundles of the neuron processes, right? And we have nerves for the peripheral nervous system. So that's as far as we differentiate the central nervous system: brain, spinal cord, okay? Then we have the peripheral nervous system, okay? and it goes into sensory and motor, right? Or afferent and efferent. So in the last couple of videos, I've said my device to remember efferent is I'm exiting the central nervous system. So I'm exiting the central nervous system. I'm going from my brain somewhere else. So I'm going to a muscle, to an effector, okay? So these are motor, right? Usually they're telling us to do something, and that it transmits impulses from the CNS to the effector organ, muscle, or gland, okay? And then we have sensory, right? We're sensing something. We're going to the central nervous system, right? So we have somatic sensory fibers, which convey impulses from the skin and skeletal muscles, right? Somatic is our voluntary control. And then we have visceral sensory that conveys impulses from our organs, right? This is our stomach growls and we notice it and we're hungry. And then we know we're hungry, okay? And then somatic is, I would like to move my arm, right? So I'm telling myself that I would like to move my arm, and then I motor, efferent, and then I move, okay? So then our motor divides into two more, right? We have the somatic and we have autonomic. Autonomic is involuntary, right? This regulates smooth muscle like organs, cardiac, so your heart, and your glands, right? We don't think about this, okay? We don't tell our pancreas when to secrete things. We don't tell our hypothalamus when to secrete things or when our heart to beat, right? These are the automatic systems. Okay, and then we have the somatic, right? Somatic and somatic. These are our voluntary movements. These are voluntary nervous system, skeletal muscle movements. Okay, so it's, oh, he's watching a video. Sorry, I was really concerned. This is when we're telling ourselves to move. I know I'm being redundant. I just want you to understand because there's a lot going on. Okay, so then in our autonomic, things that are out of our control, we have parasympathetic and sympathetic, right? I say sympathetic is your body feels bad for you, or it's sympathetic that you're gonna die, right? So it's trying to help you. It's that fight or flight, right? We dilate our pupils so we can see better. We increase our heart and respiration rate so that we can do more work to run away faster, right? And then parasympathetic is that rest and digest. It's the, let's chill on the couch. Let's not use a lot of energy. Let's store for times when we might need it, right? Like the sympathetic times. Okay, so then what are the two cell types found in nervous tissue? So we have neurons and neuroganglia. Neurons and neuroglia, right? So we have the neuron itself, right, which is the neuron, and then we have the neuroglia, which is the wrapping or the coating, and that it's like that. It's the packing peanuts, okay? And it surrounds and it supports the neurons. There are 4 cells in the central nervous system, right? We talked about astrocytes, right? So this is central nervous system, not peripheral nervous system, okay? And these are neuroglia, right? These are the ones that are supportive. Astrocytes, microglial cells, ependymal cells, and oligodendrocytes, okay? I think you need to know what those do, right? The astrocytes are abundant, they're the support, and they migrate, and they have migration of young neurons, okay? Microglial cells, touch and monitor neurons. They migrate toward injured neurons, okay, and they get rid of and destroy pathogens. So these are our immunoneurons, right? They make sure that we're in check for anything that's in there, or if we need to phagocytize anything that we don't need anymore. Then we have ependymal cells, which are ciliated. They line those cavities and they regulate the composition of cerebrospinal fluid, right? They secrete that membrane, they secrete spinal fluid, and they secrete things out into the environment, okay? And then we have oligodendrocytes, right, which form that insulating myelin sheaths, right, what we talked about in session 17, right? And those that create those nodes of Ranvier so that we can transmit impulses faster. All right, and so now we only have 2 neuroglial kinds of cells in the peripheral nervous system, right? We have satellite cells and we have Schwann cells. Satellite cells are similar to astrocytes, right, which means they're abundant, they support and they guide and they migrate. And then Schwann cells form myelin sheaths, and they're vital to the regeneration of damaged nerve fibers, right? So these are pretty much similar to oligodendrocytes. So what's the function of a neuron? Why do we care? So the function is we conduct impulses, right? It's our structural unit of the nervous system, right? Just like we said that sarcomeres were our structural unit of the muscular system, neuron is our structural unit of the nervous system. And its whole purpose is to conduct impulses. So we can send an electrical signal maybe from our brain all the way to our finger or our pinky, right? Maybe we're drinking something and we want to be real proper, and the whole purpose is my neuron wants to tell my hand that I want to do this. Okay? The whole purpose is to conduct impulses. There are characteristics for these. Extreme longevity, right? We have these neurons our whole life, and if we lose them, we lose them. That's why we have diseases like Alzheimer's and things like that, right? They have extreme longevity as long as they don't get damaged. They're amitotic. They don't divide. We have as many as we will have in our entire life when we're born. And then they have a high metabolic rate, right? They require continuous supplies of oxygen and glucose. They require continuous energy and oxygen to do that job. All right, neurons are made of a cell body, right? The soma or perikaryon, y'all learned that in lab. It's pretty much the nucleus part. It's the part where all the dendrites branch off of. And that produces and synthesizes things like ATP and protein. And then we have processes. There are arm-like extensions. We have dendrites, right, where we have inputs, right, so things come in. And we have axons, which are the conducting regions that generate impulses and transmit them along the axolemma. So the dendrites, sorry, NCAT ads. All right, so in our neuron, okay, perikaryon, right, or soma, it's the cell body, okay? These dendrites, right, here and here. And then we have this middle part that's our axon, right, which is where the impulse travels. So it would go like this, right? We get an information from here. It would go down here, right, into the perikaryon. And then once it reaches a threshold at some point, it will allow us to fire an action potential down. And then maybe it's going here and this goes to our leg, okay? But it's lots of branches of lots of neurons connected to each other that allows us to send information anywhere at all. Okay. So then we have, what is a myelin sheath? A myelin sheath is protection and an electrical insulator of an axon, right? So it literally wraps around and protects and insulates it, and it increases the rate of impulse, right, which allows faster depolarization. So pretty much what I talked about the other day is if this is our axon, right, and this is where we're getting into our soma or our perikaryon or whatever, it's our nucleus. Whoop, really pretty, obviously. Okay. And then we have, oh my, undo. We have these myelin sheaths, right? They kind of look like little jelly donuts that sit here. They protect this, right? So they protect their fat protein pads, so they protect from things, and they allow the increased rate of impulse. So that means when information comes down here and we're going to fire our axon potential to send our electrical impulse, we're going to go here, we're going to stop at this myelin sheath, and we're going to jump. And then we're going to jump. And then we go again, right? These little gaps are called nodes of Ranvier. Pretty much, it's just saying because we have to jump, our signal's going to travel faster. It travels more distance faster. Okay, so that's the purpose of a myelin sheath, right? So we have myelination in the central nervous system and the peripheral nervous system. In the peripheral nervous system, it's by Schwann cells, right? They form segments of myelin sheaths that's separated by nodes of Ranvier, which is what I just drew you, right? That's our peripheral nervous system. However, in our central nervous system, this is done by oligodendrocytes, right? And they wrap multiple axons, right? And we talk about white and gray matter. In session 17, we talked about that. So originally your brain matter is gray, right? These loads of axons or dendrites or somas are gray in coloring. However, when they get myelinated, they turn white because it's a fatty protein substance. So then that creates white matter and gray matter. So we have gray matter, which is non-myelinated, and we have white matter, which is myelinated in the central nervous system, so in your brain and your spinal cord. Okay, so neurons are classified in two ways. They're classified by their structure, right, however many processes they have, and their function is where the impulse is traveling. Okay, right? So I said structure and function. So we have structure and function, right? So these are structures. So we have multipolar, unipolar, and bipolar. So multipolar have 3 or more processes, right? There's way more than 3, right? They're most common in our central nervous system, and they have 1 axon, okay, and lots of dendrites. Unipolar, we have 2 axons, right? We have one here and we have one here. They're peripheral nervous systems, right? So they usually meet up with the central nervous system, but they're peripheral neurons. Then we have bipolar, right? So we have 2 processes right here and here, but we only have 1 axon. It only goes one way. Okay. Now these are rare. Okay. So then for our function, right, we have sensory, motor, and interneuron. We either have neurons that sense things, that tell us to do things, or neurons between. So I talked about this the other day, but I will draw it again. So say this is our sensory, okay? Right, so this is our finger, and it hurts, and it's beautiful, right? Nope, I picked orange again. And then we have this interneuron, okay, with its tells our central nervous system neuron, right here in blue, right to our brain. I don't think I can draw a brain. I think it might be better if I just like try and write brain. Whew. Stellar. Good, good. So this is our cool. So we have sensory, motor, and interneurons. Okay, so we have we have neurons that sense things, and they go from our sensation to our central nervous system. Right, we have ones that go from our central nervous system to our effectors. Right, those are our motor ones, and then we have interneurons, which are the ones between. Okay. All right, what are nerves? And they're different kinds. So they're cord-like bundles of axons. So they're just bundles of those long axons that ends up making a nerve, right? So we have sensory, motor, and mixed. We have impulses that go towards the central nervous system, right? Afferent. We have away from the central nervous system, motor. And then we have mixed, which include a little bit of both. Despicable Me is now on Netflix. Okay, so now we have where's the endoneurium, where's the perineurium, and where's the epineurium. So if you are one of my humans that sticks with me on our prefixes, endo, right, which will tell you this encloses the axon in individuals, right? So it's the smallest, right? Peri, right? This will tell us that it binds bundles called fascicles. If you remember fascicles from the nervous system, it's very similar. And then we have epi, right, which is outside or the perimeter, and so it does the entire nerve. So this is the biggest, this is the smallest, and this is in the middle. Okay. All right. So in talking about our physiology, right, our voltage, what is voltage, and what is our resting membrane potential? So we talked about yesterday that voltage is a so over here. Or I can put this right here. So that voltage is a measure of potential energy generated by separation of charge. So if I have a lot of positive charge right here and a lot of negative charge right here, I have a high voltage because they're attracting each other and there's space between them, and there's high potential for something to happen, okay? Because there's a lot of separation of charge. And our resting membrane potential is negative 70 millivolts, just like in muscles. So we have kinds of channels. We have leakage and gated channels. So leakage are not gated, they're always open, right? They're leaking constantly, okay? We have gated channels. They're proteins that change shape to open and close. If you genuinely think about a gate like at Mustang or Grove or something, you put in a code and it goes from this to this. So we change shape to open and close based on if we have the right code, the right ion, or the right whatever we're talking about. We have chemically gated ion channels, right? These are gated and they have to be bound by a specific chemical or neurotransmitter, right? So maybe it's your gate like out at a ranch, right? And so instead of there being lots of codes or lots of gated channels that we could allow us to, we have one code for like one person, right, for us. So with one specific neurotransmitter, our gate will open, our chemically gated channel will open because we got that chemical, which is our neurotransmitter. We have voltage gated channels, right? These are electrical. They open and close in response to membrane potential change. So if the membrane potential changes and we notice that something is going on in the cell, then these will change based on that scenario, okay? So there's a video in the, there's a video in the playlist for helpful videos for exam 4. And there's a video by Osmosis, and it's like a med school dude, and so it might be a little bit too much, but they do a really, really good job of showing you when the sodium channels are open, when the potassium channels are open, and what's happening at each very step. Okay, so if you're confused on that, go watch that video. And then we have mechanically gated channels, right, which open and close based on physical deformation. So if something goes out and we have a physical change, where it says change or structure determines function. So if we change our structure, our function changes. We're having that physical change that allows us to change. And I said change like 5 times. How are potentials established? So difference in ionic composition from intercellular to extracellular. So pretty much that just means the difference of ions from the inside of the cell or the inside of the axon to the outside of the axon. So the difference, okay? So there should be more sodium outside the cell, and there should be more potassium inside the cell, okay? So the difference in membrane permeability, so it's more permeable to potassium. It more diffuses out than sodium, right? So this has to do with our sodium-potassium pump, okay? And so it's 3 sodiums for every 2 potassium, and that has a lot to do with our resting membrane potential and keeping things in the right area so that we don't have a triggered action potential that isn't needed, right? What is the difference between depolarization and hyperpolarization? So let's do my little graph. Okay, so this is negative 70 millivolts right there. Ooh, okay, that's 70. Ooh, I can do this. Negative 70 millivolts. So we're like, it's time for the action potential, right? So we finally reach, so maybe this is our threshold, okay? So we finally have enough change in our voltage, right? Because this would be voltage. We finally have enough change to the voltage that we end up getting to this threshold potential, and then we can fire an axon potential, which is going to look like this, okay? And I'll talk about this in a second. So we are, as we go up, we are depolarizing, right? Because we're getting closer towards this hypothetical 0. That's way, way up there. So, right, so this is depolarizing, right? And this is when we fire that action potential. And then afterwards, we realize that we need to come back down to where we started, okay? So this is repolarization, okay? Because we want to get back down to that resting membrane potential so that if we needed to fire fire again, we could. So depolarization, when we go back down, we try to get to normal. However, this little bit, this dip, right? So we see how this would make us go back. That's where this line is supposed to be. It's supposed to be here. We'll go back to resting membrane potential. If we go below resting membrane potential, this is hyperpolarization. We've become too polarized. We've become more negative than we needed to, okay? So depolarization is the change in membrane potential where we become less negative, where we become less negative, and hyperpolarization is when we become more negative than we need to be, okay? So if you need to draw that graph, do it. What is a graded potential? So it's a potential that's short-lived and it's localized, right? And so this is more of like if you get general or if you get localized anesthesia, right? Maybe you get a tooth pulled and you get anesthesia right where that tooth is pulled. It's short-lived, it's localized, and it doesn't affect your whole body. However, an action potential or our general anesthesia, maybe you have to have surgery, and so this is a long distance, a long time, and it's our whole body, okay? So an action potential is a long distance neural connection, right? So this is like calling somebody versus it's like calling somebody really far away, right? This only occurs in muscle cells and in neurons. So there are 4 phases of our action potential. We have resting, where we're kind of hanging out, we're at our resting membrane potential and everything is good. And then we suddenly start to get some change in ions. So we get sodium that comes in and we're getting less negative and we're going up. And then we reach a threshold and we fire, okay? Remember? And then we're like, all right, well, we need to come back to resting so that we could do whatever we needed to next time. So we go down, so sodium comes out and potassium comes in, and we return to that resting state. However, hyperpolarization is when we go down too far, right? We go down here when we don't want to be that far down, is we get more negative than we'd like to be, okay? I do think you need to know which channels are open and what's going in and out, but I'm not going to go through that specifically with you. I think y'all are very capable. Okay, so what are the factors that affect the rate of propagation, also known as how fast something can travel or how fast an impulse travels? So the axon diameter, right? So the bigger the diameter, the faster the impulse, right? So think about like traffic or a highway. The bigger the highway, the less traffic there's going to be, the faster you're going to get to your destination. The degree of myelination, right? So how much myelin is on it, if at all any. And so we have two types of that. We have continuous conduction, which is slow and where we'd have no myelination. So we're kind of just going real slow. We're driving a tractor. And then we have salutatory or saltatory conduction, which is where we have myelination and it's faster, right? We're driving a Ferrari. Okay. What are the two types of refractory periods? Okay. So a refractory period is a point where a neuron cannot trigger another action potential. So we cannot send another action potential. And that's because our membrane potentials, right? We're not back at that resting membrane potential. And so we can't change the concentrations because we don't have enough in the regular spot to even change any. We don't have enough difference. So we have absolute and we have relative refractory period. So I like to tell you like this. So absolute refractory period is right when you've done a bunch of homework or you've taken a test or something and you just need a break. Your brain really needs to rest and chill and you just need some like time to breathe and recoup, right? Which in the neuron is we're putting all the charges back, everything's kind of going back to normal and we just need the time to do that. A refractory period or relative refractory period is when, okay, and spatial is when we're stimulated simultaneously. So temporal is 1, 2, 3, 4, right? They're happening quickly. Spatial is they're happening so fast you can't even tell that they're different. So it's really, really fast. It's simultaneous. They're all together, right? Okay, it's almost like how a video is just like a bunch of pictures, right? But I could take like 4 or 5 pictures like really close and you could tell that they were different pictures, right? But a spatial would be, it's a video. Okay, they're happening so fast you can't even tell that they're pictures. Okay. What are the neurotransmitters? Why do we care? They're the language of the nervous system. They're pretty much the letters of English for the nervous system. This is how we talk, how neurons talk to each other, how our nervous system talks to each other. Okay, so the effects are we have excitatory, right, where we depolarize so we can have an action potential, or we have inhibitory where we hyperpolarize so that we're all the way down here and we can't even create an action potential. All right. So we have actions. We have direct and indirect. So direct is we bind to an open ion channel, or we have indirect, which doesn't cause an immediate reaction. We don't bind. It acts through a secondary messenger. So maybe it doesn't bind, which makes this other thing happen. Okay, so it's an indirect relationship that's happening. So in the central nervous system, we have white and gray matter, right? We said gray matter was non-myelinated. It starts off gray. We have white matter, which is gray matter that's myelinated in the central nervous system. That myelin is white because it's lipid-based. We have ventricles, which are pretty much fluid-filled chambers of spinal cord fluid, and they protect the spinal cord, and they're filled with cerebrospinal fluid lined with ependymal cells. Ependymal cells' function is to secrete cerebrospinal fluid. Okay, so we have gyri, I don't know. They're the ridges of the hemisphere. We have sulci, which are shallow grooves or divots, okay? And then we have fissures, which are valleys. And these are just on your brain. I don't think I've ever seen them test these. Don't know. All right, so parts of our brain. The cerebral cortex. This is our executive function. This is the man, right? Or woman, or whoever is in charge. This is the site of our conscious minds where there is all inner neurons. It's all firing, but we're all between, and there's no axons, right? Executive suite. This is the man that makes the choices, okay? So we have a motor area, a sensory area, and an association area. Motor area, voluntary movement, okay? Sensory is conscious sensations, right? So we're sensing things. And then the association is our integration, right? Those are the same three things we've been talking about since the beginning of this. So those are always pretty much our big three functions. So we have contralateral and lateralization. Our eyes are contralateral. If even our brain hemispheres. So if you look this up, it's crazy. So this eye is seeing stuff that's over here on the opposite side. So my vision from here goes here. It goes back to the other side of my brain. Let's just look this up. I know we're kind of in a rush, but let's do it anyway. Contralateral, this is crazy. Watch a video on this. So, right, so the opposite side of the body is processed by here. So, your left hand is perceived by your right hemisphere, okay? Your right hand is perceived by your left hemisphere. Same thing with our vision, right? This right eye perceives lots of vision from the left, okay? So we have parts of our brain that interpret stuff associated with the opposite sides of the body. However, we do have lateralization, which is specialization of hemispheres. So we have right and left brain, right? So they are different and they do have different functions. So we have that lateralization where it's laterally, so we have one section and the other section, but contralateral means things from one side are interpreted by the other side of the body, okay? There are 4 regions of the brain. We have the superior part, so we have 3 layers. We have the cerebral cortex, white matter, and basal nuclei. These are the cerebral hemisphere, okay? And then we have that's one of 4 regions of the brain. Cerebral hemisphere, diencephalon, brainstem, cerebellum, okay? Our cerebral hemisphere is 3 layers. It's a cerebral cortex, white matter, and basal nuclei. The diencephalon is made up of your thalamus, epithalamus, and hypothalamus. Genuinely, I know this is all a lot of information. I would know what is in what category, okay? I would know that the thalamus, epithalamus, and hypothalamus are in the diencephalon, right? And they are relay stations, okay? They pretty much are the place where all the information is sent, and then right there they go, oh, this needs to go here. They're the filing cabinet. They're the organizers, okay? I would also know that the pineal gland secretes melatonin, and that is in the diencephalon. That is important, okay? So this includes the mammillary body and infundibulum. You learned about these in lab, or at least you remember where they are, hopefully. So we have the brainstem, right, which is all your automatic behavior, okay? This is breathing, heart rate, right, all that jazz. So automatic behavior. In our brainstem, we have the midbrain, the pons, and the medulla oblongata. The midbrain is made up of the cerebral peduncle, the corpora quadrigemina, right? And then the corpora quadrigemina is made up of the superior and inferior colliculi. Superior colliculi is your visual reflex. Inferior colliculi is your auditory reflex. I remember that my eyes are higher than my ears. So my superior is eyes and my inferior is ears, okay? And that's in your corpora quadrigemini. It's just these 4 little sacs. It's like, that's why it's called that, because it's quad, it's 4. Anyway, these are in your midbrain. In your pons, we have conduction tracts, which are structures that connect to each other, right, to send more information. And we have a reticular formation, which is a sensory filter. We filter all sensations through this reticular formation. Lastly, we have the medulla oblongata, which is our first enlargement of the spinal cord. You know what the medulla oblongata looks like, hopefully. Look it up, it's cool. This is your automatics reflex center, right? So it's cardiovascular, respiratory. We don't have to tell ourselves to breathe. We don't have to tell ourselves to have our heartbeat. That would be insane. The medulla oblongata is the thing that does that. They talk about in the parts of the brain how if you're going hindbrain, right? So this is hindbrain, and then this is forebrain, right? So frontal, and then this is hindbrain. As we go farther back, we get more primitive. So this is breathing and heart rate and things like that. And then we get up here and we get like voluntary control, right? And then we get like emotions and feelings and thoughts and integrity and who you are, right? So these are more evolved later and more complex functions. And then as we go back, right back here, we get more things like medulla oblongata, which are like innate things that we need to live, okay? We're primitive. So then we talk about the cerebellum, which inputs information from the cortex, right? It provides receptors for muscles and plays a big role in balance. They like to ask about the cerebellum and balance, okay? So how is the brain protected? It's covered in meninges, which is 3 layers, right? We have a dura mater, arachnoid matter, and pia mater, right? These pretty much just cover the brain. They're just layers of covering on the brain. So, and then we have, so it's also protected by cerebrospinal fluid. It's a cushion. It nourishes the brain, and it has phagocytic stuff in it to make sure that we are clean. We also have a thing called the blood-brain barrier. I'm sure you've heard of it in some medical show or something. It's an impermeable barrier that keeps away lots of things in our blood, right? If we go septic, right, and we get something in our blood, we get bacteria or we get something in our blood that we don't want in our brain, we have this blood-brain barrier to make sure that stuff from our blood doesn't go into our brain and infect our brain, okay? It's almost like a mesh, kind of like a strain in a very, just as an example. We can do this. We're ready. We got this. Cool. All right. So what is the significance of the spinal cord? It provides a two-way communication from the brain to the body, right? If we just had the brain and we just had the body and we had no spinal cord, they're not communicating. We have to make sure we have a way to communicate. We need a way from get to point A to point B. All right. We have reflexes. We have rapid reflexes, right, that occur automatically, right? So when people hit your knee, or that funny bone feeling, right? Things that are reflexes that we don't actually get to control, okay? And then we have pathways called reflex arcs, okay? So reflex arcs are, okay, right, where we receive stimulus and it goes up to our central nervous system, it's integrated, and then we have something that is stimulated. So, but it's just the pathway for reflexes. A reflex arc is just the pathway of neurons up to your central nervous system to tell you what the heck to do for a reflex, right? So when the doctor hits your knee with that little thing, right, the reflex arc is activated and it does all this jazz to tell you to kick your knee out, okay? There are 2 types of reflexes. We have monosynaptic and polysynaptic. So monosynaptic is when we have no association of neurons, no integration, a real reflex. There is no controlling that. It just occurs, okay? And a lot of that's based on ligaments and placements and stuff. A polysynaptic reflex is when at least 3 neurons are involved and it includes integration. So this reflex, it goes, maybe I get hit in the shoulder, right? Or whatever. And then it goes all the way out to my brain, tells me what to do, and then comes back, right? So it involves at least 3 neurons and it includes integration, which means our brain is having to understand what is happening, okay? So now we're going to talk about the peripheral nervous system. So what is the difference between sensation and perception? So sensation is the awareness of changes, okay? So sensation. This has a little bit of coffee in it. If I put more coffee in it, the fact that I could sense that there is more coffee in it by its weight is a sensation, right? Whereas perception is the interpretation of those stimuli. So that would be, oh, I thought I had less coffee than this, right? And I'm thinking about this. I'm integrating that information, okay? That's a perception is what I'm thinking. So then the difference between transduction and adaptation. So transduction is when energy stimulus is converted into a graded potential. So we're transducing that. So we're going from energy into a graded potential. So we're transducing, like transduction of some kind of stimulus. And then adaptation is a change in sensitivity to a constant stimulus. Like when you walk into a room and it smells really weird or really nice or something, or it's really cold, and then after a while it goes away, okay? Or the fact that you don't see your nose, okay? If you look at your nose for a while, okay, you're gonna be like, oh, it's right there. But after a while, you don't see it anymore. So we've adapted. Our sensitivity either goes up or down based on how long we've been there. And so that's why you can't smell yourself, or maybe you can't tell that it smells really nice in here, or you can't smell the broccoli you just cooked, now it smells like farts in your house, all right? So what are the sensory receptors? I have a feeling you'll all be good at this. We have mechanoreceptors, right? Those are mechanical force, okay? Thermoreceptors, temperature. Photoreceptors, light. Chemoreceptors are chemicals, and nociceptors might be the only one you haven't heard of, right? This is pain, right? This is stimulus of different kinds of pain, okay? And so then we have places for those receptors to be. We have external receptors, internal receptors, and proprioceptors. External receptors respond from stimulus outside the body, okay? Internal receptors, inside, stretching, temperature, chemical change, fever, right? And proprioceptors have to do with our body movements, right? Maybe, right, muscles, joints, tendons. Maybe we've been stretching a really long time and we're propriocepting. Our proprioception is now it kind of feels good. Or if you ever tried to balance where you close your eyes, proprioception, that's proprioreceptors, is you can tell a lot where your body is and how your body movement needs to be, your balance, if your eyes are open, because we're allowed to assess the situation. But if you close your eyes and try to do it, this is a lot of proprioception because you're having to integrate that information without a visual stimulus. Okay. So then we do vision. We are almost done. Everyone take a deep breath. Eyebrows, okay? So these shade our eyes from the sun. They actually have purpose other than being pretty on our face, right? They shade our eyes from the sun or from sweat reaching our eyes. If you ever worked outside or been working out or done some like hot yoga, you'll notice that you don't get a lot of sweat in your eyes. And that's because your eyebrows are protecting your eyes. They have actual purpose. Your eyelids protect from up here, right? Our eyes are really sensitive. So we want to make sure that they're pretty much contained. They're contained and they're protected. Our conjunctiva, whatever, is the transparent mucus that lines the eyelids. So it's like that inside piece of your eyelids that's a mucus layer and it protects. The lacrimal apparatus, you remember your lacrimal piece in here in your skull, right? It releases tears and ducts that drain into your nasal cavity, which is why if you're like me and you cry for like maybe 4 seconds, you can't breathe out of your nose at all, okay? So it drains into your nasal cavity, which ends up stuffing your nose up, okay? So the extrinsic eye muscles are muscles here and here that you probably have never seen a picture of, and it allows you to move your eyeballs all around, right? It allows you to roll your eyes when you learn how to do that when you're 12, and then your parents like, never do that again. Okay, I like always roll my eyes like just for funsies because I feel like it's something you don't get to do a lot. And my parents always like, are you rolling your eyes at me? I'm just rolling my eyes. Anyway, that was weird. Sorry. What are the 3 layers of the eyeball wall? So we have an outer layer, right? It's an avascular layer. It has no blood flow. We have the sclera and we have the cornea. The sclera is the white of the eye. It protects and shapes and allows that muscles to attach so for us to move our eyes. And then we have the cornea, which allows us to receive light. So if you looked at an eye sideways, okay, you would see like this big bulge of kind of like a clear, it looks like the lens, but it's not. And it's the cornea, and it literally allows the light to come into our eye, which is why it's clear. So then we have a middle part, right? Right, this is our pigmented layer, right? We have our choroid, I think so, that supplies blood, right, and absorbs light and prevents it from scattering, right? Look up the videos on the eye that are on the YouTube, right? Definitely go look at all the structures. Those are really important, and I feel like it's hard to know those without seeing them. The ciliary body, which controls the shape of the lens, that literally it looks like a little bunch of little hairs, and the lens is in the middle here, and it will pull and stretch it based on how much light we have, right? So, you know, when you wake up in the morning, you're like, wow, it's really bright. Or if you've ever done the thing where you look at your eye and then you turn out the light and then you turn it back on, you'll see your pupil shrink and enlarge, and your iris allows a lot of that light to come in and out, okay? So then we have our inner part of our eye, which is our retina. And it's our innermost layer. It contains all these photoreceptors, which allow us to see things like color and how much light is going on. So we have our pigmented layer. We have our neural layer. Okay, so The neural layer has the optic disc and the blind spot and the cones, right? So hopefully you know what a rod and a cone is, right? So cones, C for color. So they have colored sight. They allow us to see different kinds of colors, right? This is a low sensitivity to light, and it requires bright light for activation. So if you've ever noticed how at night when you're driving or in your room when you're going to bed, you don't really see any color. You see a lot of black and white, and that's because there's not a lot of light, and so our cones are not activated. We have 3 pigments, by the way. It's red, green, and blue, and then everything else is just a mixture of everything. And then we have rods, which are really sensitive to light. These are best for night vision. These are your black and white ones, okay? This is why at night when you can see in low light, okay, you see lots of black and whites because your rods are what's activated right then. So there are chambers of the eye. There's a posterior and an anterior segment, right? You know where the posterior and the anterior are. So the posterior is that vitreous humor. It's that clear jelly, kind of looks like if you were to cut open a grape, I would say is what the vitreous humor looks like, is that jelly stuff that makes up the inside of your eyeball. It allows us to transmit light through it, which is why it's clear. And then it allows pressure in the eye, right? Because it's pretty much the air in the balloon of your eye. And then we have the anterior segment, which is divided into the iris, aqueous humor, and it has 2 chambers in it. We have an anterior and a posterior chamber. Okay, so it's the anterior chamber is from the cornea and the iris, and then the posterior chamber is between the iris and the lens. Just recommend looking these up. I know it seems like a lot. Just look it up. So what is phototransduction? It's the process by which light energy is converted into a graded potential by photo and bipolar cells. It's the fact that I'm looking at this light right now, and it can go into my brain, tell the rods and the cones and whatever needs to be told that I'm going to have a graded potential, and I fire an action potential to allow those photoreceptors and bipolar cells in my brain to be like, wow, that is bright. So I'm taking the energy and the light and creating an electrical stimulus out of it through those photoreceptors and bipolar cells to tell myself that it's light, right? Because I'm not just seeing light and being like, oh, that's light. I'm using that energy through here to understand that that's what's going on. So then we have our visual pathway. Oh, my neck hurts. They always ask this. In my opinion, they always ask the path of light. I would just memorize this. Okay? Not so much this. Okay, the path of the impulse. I don't really want to talk about that. I don't feel like that's highly covered. Right, so then we have parts of the ear, right? We're talking about the ear now. We have the external ear, which is literally this part. It's the auricle. It funnels the sound in. We have the external auditory acoustic meatus, which is literally your ear canal, right? That's where you're putting your Q-tip. Don't, hurt your ears. Anyway, transmit sound into, right? So we're funneling. We get sound in there. There's also a video on the ear. And then we have that tympanic membrane, which is literally your eardrum. You will know what that is if you've ever accidentally hit it with a Q-tip. Don't do that, okay? And then we have the ossicles, right, which are the malleus, incus, and the stapes. They're the 3 smallest bones in our body, and they're actually in the ear, and their purpose is to allow that sound wave to travel through the eardrum and vibrate the malleus, incus, and stapes and turn that energy, that mechanical energy of vibration, into an electrical impulse to tell our cochlea and things in the inner ear that we need to transmit that electrical signal, okay? The inner ear. Okay, the inner ear, we have a vestibule, right, which is for our balance and linear movement. If you're anything like me, I'm really bad at riding roller coasters, and I can't read in the car because if I'm looking down too long, my inner ear movement, right, my vestibule will move and it will make me car sick. Okay, so it has a lot to do with our balance and linear movement. We have the semicircular canals, right, which is a lot of our rotation of our head. We know where our head is in space because of the way that fluid moves in the semicircular canals. And then we have the cochlea, where sound waves are transmitted into a neural impulse, right? So we go from sound wave to mechanical, right? And then we go to a neural impulse that moves these little hair cells. They literally bend them and make it a mechanoreceptor. So we understand that the hair cells get bent, and that sends a signal, okay? The pathway of sound, they always ask this. Always ask this. They really like to talk about the vestibular apparatus and the semicircular canals having to do with your equilibrium and your balance and your linear movement, like how fast forward or backwards am I going, okay? This is a lot of roller coaster stuff, right? If you've ever been on, I think it's Mr. Freeze, where they throw you back and you're like, oh my gosh. That has a lot to do with your our vestibular apparatus and our vestibule, where we're sensing our linear motion and how fast and our balance. Okay. Chemoreceptors, right? So we have chemoreceptors for our nose and things. So we have smell, which is olfaction, and taste, which is called gustation in our very sciencey terms, right? They're only excited by dissolved chemicals in the air and saliva. So those chemicals in the air and saliva and your saliva are what allow you to smell things or taste something and have those nuances of smells and tastes and things that go through our nose and through our mouth to excite things based on their chemicals. We're not going to go into all of that. Olfaction is your sense of smell, and it also includes temperature and pain. So you can tell if something's hot or cold, and you know that if you plug your nose and then you try and eat something, that your sensation is much more dulled, because you need those things, those olfaction and gustation together to really create that whole picture. All right, gustation, right? We have taste buds, right? Those are on your tongue. We have taste receptors and little hairs that do a lot of what we do in the ear, but with taste. And then you have your 5 basic sensations, 5 basic tastes, which is sweet, sour, salty, bitter, and umami, which is that savory kind of meaty flavor like garlicky. Sorry, I'm just talking a lot. Okay. Session is in 5 minutes, so I'm going to log in and let this render, and then I'm going to upload it, and we're going to have Q&A. Please shoot me a text or a reminder, anything you need to talk about, anything we talked about. I know I kind of went fast. Please use the videos on the YouTube and the playlists and everything like that. I think that is what's really going to strengthen your knowledge and let y'all to do good. I will see you all literally in 5 minutes, so you don't know that. And then I will see you all again later. Let me know.